Okay, so our first exam question states, in the process of semi-conservative DNA replication, the two strands within a DNA molecule are separated. Each then acts as a template for the formation of a new complementary strand. Describe how the separation of strands occurs. Okay, so we've looked at this already. How does the separation of two DNA strands occur? Okay, feel free to have a go at it. I'm just going to be going through it of how we can answer this question in relation to the mock scheme. Okay, so basically, how does the separation of strands occurs? Well, what happens in step one? We had DNA helicase, right? DNA helicase breaks the hydrogen bonds between the complementary base pairs of DNA. That's the sentence I told you to remember, and that's basically what I'm going to write. Okay, so I'm just going to write DNA helicase breaks the hydrogen bonds between the complementary base pairs of DNA. Okay, that's how I would answer this question. And if I show you the mark scheme, it says DNA helicase, that's 1 mark. And then it says breaks the hydrogen bonds between base pairs or complementary bases. That's also 1 mark. Okay, and I've literally got 2 marks by just writing one sentence because that's the one I've told you to memorize. DNA helicase breaks the hydrogen bonds between the complementary base pairs of DNA. Okay. Next question, it says describe the role of DNA polymerase in the semi-conservative replication of DNA. Okay, so remember, DNA polymerase is part of the final step. What does it do? Well, it joins the adjacent nucleotides together. And remember, what did I put in brackets? I put in condensation reaction and I put phosphodiester bond. Okay, so DNA polymerase, how I would answer this, DNA polymerase DNA polymerase catalyzes the formation of phosphodiester bonds. So catalyzes the formation of phosphodiester bonds through a series of condensation reactions. Okay, that's how I would answer this question. Okay, because in my sort of template answer that I had, it had DNA polymerase, it had condensation reaction, and it had phosphodiester bonds. Okay, so that's, if a question like this came up, I would literally follow that path and include everything. Okay, just to make sure. Okay, so joins adjacent DNA nucleotides. So we've actually missed that point because I think I was 2 on the condensation reaction and phosphodiester bond, but there are 3 marking points. We've got condensation reaction and we've got phosphodiester bond. Okay, this is also a very important point that I've missed out there. It joins nucleotides. That's essentially what it does. It joins adjacent DNA nucleotides. Okay, but I had the other 2 points because it was written down and then you had to include it. I've just forgotten the first point. It joins the adjacent DNA nucleotides. Okay, so joins adjacent DNA nucleotides. Okay. Next question, it says scientists replicated DNA in a test tube. To do this, he mixed an enzyme with identical single-stranded DNA fragments and a solution containing DNA nucleotides. Name the enzyme used in this DNA replication. So what is he doing? He is replicating DNA. Okay, he's replicating DNA in a test tube, and he mixed an enzyme with single-stranded DNA fragments. So in the test tube, we have these single-stranded DNA fragments. We need to make them double-stranded. How do we make them double-stranded? Well, we need DNA polymerase to catalyze the formation of phosphodiester bonds between adjacent nucleotides through a series of condensation reactions. Okay, so it's going to be DNA polymerase. Because they're already single-stranded, we don't want to break them even more. We want to actually catalyze them joining together. That's why it's DNA polymerase. Okay. Now it says, give two features of DNA and explain how each one is important in semi-conservative replication. Okay, this question leads on from our last lesson. Okay, we looked at this in our last lesson, and if you don't remember, I did sort of send out bullet point list that you do need to remember. Step one, well, not step one, point one that I would make is that it has two strands, so each of them can act as a template in the semi-conservative replication of DNA, because that's the structure of DNA. So two strands, so each can act as a template. Okay, that's always the first point that I write down whenever a question like this comes up. And I also write down the complementary base pairing rule, because complementary base pairs allow accurate replication. Okay, those were the 2 points that I would have. Weak, easily broken hydrogen bonds. Okay, yeah, so this is another point that you could have had. If I just get rid of. Okay, it says two strands, so both can act as a template, which we had. Always my go-to point. It will be there on every mock scheme. Complementary base pairing allows accurate replication. That's always on my go-to as well. Okay, and that would be worth 2 marks. Okay, final question. It says describe the role of two named enzymes in the process of semi-conservative replication of DNA. So this is the model answer that you need, basically. And I'm not going to attempt answering this. I'm just going to show you the model answer. This has to be on a flashcard. This is basically the process summed up. Okay, so if you have a flashcard, if you're making flashcards, this mock scheme answer must go on that flashcard. Okay, DNA helicase causes the breaking of hydrogen bonds. Okay, we need helicase. Don't say hydrolyzes hydrogen bonds. Okay, because they will always reject that. They don't hydrolyze hydrogen bonds. They don't release water. We know that the formation of them is using condensation reaction. However, that's between the adjacent nucleotides. It's not interfering with the hydrogen bonds. So the water doesn't actually come from the hydrogen bonds. Okay, DNA polymerase joins the DNA nucleotides, forming phosphodiester bonds. Okay, and we've named 2 enzymes. This is basically the process summed up in 3 points. DNA helicase breaks the hydrogen bonds between DNA strands. DNA polymerase joins the DNA nucleotides, forming phosphodiester bonds. Okay, that's basically what happens for 3 marks.